The first of our biology videos, which is monoclonal antibodies. I'll put in the description who it's suitable for, but I hope you enjoy this one. So this comes at the end of topics like infection and response, and it's one of those topics that goes up to level 9, 8 or 9, so it's important that if you're aiming for those grades, that we know what it's all about. Okay, so first off, a quick reminder, can you remember what antibodies are and what they do? So if at any point you need to pause and have a think, that's absolutely fine. Pause now. If not, let's get into it. So first of all, what they are and what they do. So it's really important in biology, as you know, we get the language correct. So this is what a question at this level will be looking for. So an antibody is a protein. If my pen, there we go, is a protein. And it's produced by lymphocytes. Now, lymphocytes are a special type of white blood cell in response to and counteracting a specific antigen. And we'll look at the difference between antibodies and antigens in a minute. Okay, they are part of the immune system and they destroy pathogens. Remember, we don't have any fighting off of pathogens. Destroy, digest, kill, absolutely fine, but no fighting off that have entered the body. Okay, so just in case you're not sure, an antibody looks this kind of shape. I'm going to do it up here in the corner. A Y shape like this. So they will always be represented by a Y shape. So you can see that they are antibodies. They're made of proteins. They're actually made of 4 different proteins, but for GCSE, we just need they're made of proteins. And it's this area here, this area here, love technology, this bit here, so this V-shaped bit, that is actually complementary to the antigen on a pathogen. So complementary with an E in the middle means the shapes fit together exactly. So if we had a pathogen down here, let's imagine we had a bacteria or a virus, the antigen might be this shape, okay, with the pointy bits, and the antibody maybe this shape. Okay, so they fit together really, really nicely. They are complementary shapes. Okay, let's look at the next bit. Sorry about the noise. Suggest how we might make large amounts of these antibodies. So we know what antibodies are, and this top monoclonal antibodies means making lots of the same antibodies with the same site with the same complementary shape. Okay, so mono means 1. Clonal, obviously like clone, so they're all identical. And then we've got antibodies. Now there's a specific way that we have to know how to make them. There are different ways to make them, but this is the one that the exam boards want. We need to get antibodies that are all identical. So we can't just use white blood cells directly because they can't divide. So white blood cells, once they're made, they can't divide. They've already made antibodies. However, we can use them indirectly. Okay, so let's have a look. Oh, sorry, very sensitive today. There we go. We take a mouse, okay, and into that mouse we are going to inject the antigen. And remember, the antigen is on the outside of a pathogen. So if this is our pathogen, might be that shape. So what we do is we inject the antigen into the mouse. The mouse is then going to, as a natural response, make those white blood cells, and lots of white blood cells, and they are going to make antibodies against the antigen. But those antibodies are all going to be identical, okay, because they are the complementary shape to this antigen that we've injected in. So at the moment we've got some antibodies that are the correct shape. Now, while that's going on, we are going to grow some cancer cells or some tumour cells in a petri dish. The reason we grow cancer cells or tumour cells is because they divide really quickly. And we can use them to make our antibodies divide, well, not divide, but encourage them to replicate very quickly. So once we've done that, we're going to take the white blood cell from the mouse, and we are going to take the cancer cell. And we're going to fuse them together to make this hybridoma cell, it's called. Okay, this will be summarised in a minute. So we make a hybridoma cell. Now that hybridoma cell, remember, it's got the lymphocytes that are making antibodies, and it's got the cancer cells that are replicating. Okay, so once they're together, they can start dividing and make antibodies against that antigen. And as the tumour cells are encouraging them to divide, and that means that we get lots and lots of these antibodies. Now at the moment, they're all identical. Okay, so we have made all identical. Now there's our summary page. So we're injecting the mouse here with an antigen. Okay, then we remove the white blood cells from the mouse, the lymphocytes. We're going to fuse it with our cancer cell over here, get our hybridoma cell, and the hybridoma cell can both divide and make antibodies. So it's got the best of both worlds here, against whatever it was injected against. In this case, it's a hormone. And we call them monoclonal antibodies. So these antibodies here are clones of each other. And once they're all produced, they can then be purified, okay, because obviously to make them, if we're going to use them for humans, we need them to be purified. Okay, so there's a lot of keywords in here that we're going to need. So let's just have a quick look at the uses, because they do ask you about those as well. Okay, so what we can do with those monoclonal antibodies is actually match the antigens on the pathogens to help identify pathogens. Or they can be made to stick to specific cells in the body, like cancer cells or platelets. So we will make monoclonal antibodies for a specific purpose. All right. If you're not sure what platelets are, it's there. They're little fragments of blood cells that help blood clot. But I'm sure you all know that anyway. Right, a little thing for you to do here then. Explain how monoclonal antibodies could be used to identify where a blood clot has formed in parts of the patient's brain. So I've put picture clues over the missing words. So pause the video, have a little go and see if you can do it, and we'll unstick it all in a minute and have a look. Right, hopefully you've had a go, so let's have a look. Excuse me. Since monoclonal antibodies can be made to stick to certain types of cell, they can be used in medical diagnosis. This is often done by making the antibody slightly radioactive. And when the antibodies attach to cancer cell, the radioactivity can be detected using a PET scanner, and the position of the cancer can be found. Okay, so what we've done is we've made our monoclonal antibodies, and we make them slightly radioactive, and then we send them into the body to attach to the cancer cells, and then we can use a PET scanner to see where those cancer cells are and then treat the cancer, obviously. So this is separate from the cancer cells that we use to make the monoclonal antibodies. This is like a cancerous tumour or something in the body. Alright, so that's one use. So now we need to think about kind of advantages, disadvantages. So if we use this for cancer treatment, for chemotherapy or radiotherapy, state a disadvantage of treating cancer this way. So what would be a disadvantage of chemotherapy and radiotherapy? That's, have a think, and I'll put the answer up now. They also expose healthy cells to the drugs. Okay, so just general chemotherapy. We're not talking about monoclonal antibodies now. We're just talking about general chemotherapy or general radiotherapy. It goes through the whole of the body. So not only is it destroying cancer cells, it's also destroying healthy cells in the process. So this is where we can use monoclonal antibodies, like in the last slide, to actually target the cancer cells. So that's the only thing that's treated. And here's another way. We can attach cancer drugs to monoclonal antibodies, and those monoclonal antibodies will attach to the antigens on the cancer cells, and it's only those cells that get treated. So you don't need so much drug going around your body. It kills the cancer cells, and we don't damage so many healthy cells. I don't know if you're aware of chemotherapy, but it can be, the side effects can be quite nasty because it's a very big dose of chemicals that are going into your body, sickness and things like that. Okay, doing really well. So we've got two what we call perfect answers here, or high grade answers. One is describe and one is explain. So we do this over and over again. Describe, say what you see if it's describing a graph. Say what you're going to do if it's describing an experiment, and say what happens if you're describing a method or a process. So this is a process. Describe how, monoclonal antibodies that deliver drugs to kill cancer cells could be produced. So that's that first bit that we did. And then explain, where are we? Explain. Oh, so explain is using because, why, so. So explain why using monoclonal antibodies in the treatment of cancer could be better for the patient than traditional chemotherapy or radiotherapy. So we've just talked about that. We're not killing healthy parts of the body. There's less harmful drug in the body. They're only going to attach to the cancer cells and nothing else because they're complementary shape. All right, so you get the idea. If you had a 4 marker, these are the key words that you need to be saying in these answers. All right, so very well done. Now, the question that comes up the most, other than describing how we make monoclonal antibodies, is a pregnancy test stick or another test stick that is testing for something else. Okay, so this is a pregnancy test stick. I'm sure you all remember the COVID tests. They worked in a similar way. We can test for certain chemicals in the body in a similar way. So whichever version you get, it should work very similar to a pregnancy test. So we're going to go through this one to make sure everyone understands, because it is a bit of a tricky one. So if you get pregnant, your body produces a hormone called HCG. Human chorionic gonadotrophin, I think. Don't quote me. So as soon as that embryo has implanted it, your body will start producing HCG, but it will only get to levels that can be detected probably after 12 to 14 days, something like that. And that is what the pregnancy test is testing for. These pregnancy tests have to be really sensitive because it's only a small amount at the beginning, and then as you go through your pregnancy it goes up. So it uses antibodies. Sorry, for some reason the pen isn't showing up on my screen. It uses antibodies that fit the antigen on the HCG hormone.